Dr. Anjali Venkateshwaran, and we'll be introducing today's speaker on behalf of Biomere, a preclinical CRO with locations in Boston, San Francisco, and China. Our speaker today is Dr. Michael Doe, Associate Professor of Neurology at Harvard Medical School and Boston Children's Hospital. Mike obtained his PhD in neurobiology at Harvard Medical School and completed his postdoctoral training at Johns Hopkins University in 2011. The research focus of Mike's lab is to understand how light drives visual perception and the setting of the internal body clock. Mike's talk is titled Origins of Extraordinary Visual Acuity in Primates. So, without further delay, it is my pleasure to introduce Dr. Michael Doe.
Thank you so much, Anjali. It's a pleasure to be here, and I really appreciate your gracious introduction. As Anjali said, the title of the talk today is Origins of Extraordinary Visual Acuity in Primates. And this title might strike some of you as being a bit funny. Most of us, when we're thinking about visual acuity, do so in the context of, say, sitting in the optometrist's office where we're squinting at the eye chart and hoping that our visual acuity is actually better than it was the last time. And so it can come as a surprise to hear that our visual acuity is in fact extraordinary. It is the highest visual acuity known among mammals. For example, we see 10 times more detail than a cat can and 100 times more detail than a mouse can. By our standards, these species are legally blind. So what is the origin of this extraordinary visual acuity? It actually begins at the very first step in vision, in the retina, which is that light-sensitive tissue that lines the back of the eye. Let me show you what a human retina looks like, and it's not very different from the retinas of other primates. So this is a view where you're focused on the central retina of a human being, and I want to draw your attention to this central area, which is called the macula. This pigmented region is quite special. At its very center is a specialization of the retina that is called the fovea. The fovea is something that we and other primates possess, but is lacked by all other mammals, and it is, to a large degree, why we can see the world in such detail. You might have heard about the macula before in the context of age-related macular degeneration. This is a devastating disorder for blindness. It is actually quite common. It affects 10 million individuals just in the United States. And if you look across the world, age-related macular degeneration is the third largest cause of vision loss. If you confine your analysis to developed nations, it is the leading cause of vision loss. And you can see an example of it here. This is age-related macular degeneration at an early stage, and there's a remarkably selective deposition of this abnormal substance called drusen right in the macula. In a later stage of the disease, you can see that blood vessels have infiltrated the macula and begun to bleed. And this bleeding can destroy the macula and, at the center of the macula, the fovea. Now, without the fovea, we are legally blind. We're left with our peripheral retina, but the peripheral retina cannot resolve the image at a fine grain. And that's why if there's something in your peripheral field of view, you might know that it's there, but you won't be able to make out any of the relevant details. You have to look at it and put its image on your fovea before you can see what's there to your side. So our progress towards understanding the cause of macular degeneration and towards treating macular degeneration, it's very slow. And there are two reasons that I'd like to emphasize about why we have such broad gaps in knowledge here. One is that the macula and the fovea are specific to humans and primates. So if you want to study this specialization of the retina directly, you have to do it in primates. And primates are difficult to obtain and difficult to study. What's more, even if you did have access to the primate retina, the fovea is absolutely minuscule. It's just 1% of the retinal surface area. So there just isn't that much of it to examine. I would argue that if we had a better understanding of how the fovea worked, not only would we have an appreciation for how extraordinary visual performance arises, but we would lay a stronger foundation for the earlier diagnosis and the more effective treatment of macular degeneration. So what do we know about the fovea? Let me zoom in and give you more detail on what this specialization looks like. So here's another view of the human retina, and there's the macula at the center and the fovea right here where the pointer is. And if we zoom in and look at the level of the photoreceptor cells, those neurons that capture photons and convert them into electrical signals, this is what we see. This is actually a view of a macaque retina, a macaque monkey retina that is mounted live on one of our microscopes ready for analysis. And what you see are these tiny cells that are extremely densely packed. These cells are just 2 or 3 microns in diameter, and they can't really be packed more tightly than this. These are cone photoreceptors, the kinds of photoreceptors that we used to see in daylight and that we used to see in color. And they're forming this pixel array that you can imagine resolves the world in an extraordinarily fine grain. For comparison, let's look out in the peripheral retina, where, as I mentioned before, we're legally blind. And now the cones are these very large cells, where there's a systematic progression from this ultra-fine pixel array in the fovea to this coarse array of large pixels in the peripheral retina. And these cones, being so large and so widely spaced, cannot resolve the image in sufficient resolution for us to do things like read and recognize faces as we normally do on a day-to-day basis. Now the cones are interspersed with rods. Rods are these tiny cells here. Rods are so sensitive, they allow us to see in starlight, but in conditions like this, they're desensitized and they're not contributing to vision. Okay, so if you look at it from this perspective, it makes sense. It looks like we understand how the fovea works. It's made up of these tiny cells that can pick out details. But if we just take a step back and look from a different perspective, mysteries arise. Okay, so here's another view of the fovea where we're looking at it in cross section. And you can appreciate from this view that the fovea is a pit, right? So the cones lie here in the middle, and it's thought that this pit structure actually enhances visual acuity because light has direct access to the cones. The image can be focused right on that fine pixel array. If the fovea were not shaped like this, light would have to go through layers of tissue and potentially undergo scattering that degrades the image. So the pit seems to be advantageous, but there's a consequence, which is because the cells that the cones have to talk to send visual information to the brain are pushed aside, the cone now has to extend a long fiber, an axon, in order to make contact. Let me show that to you on a larger scale here, and let me explain to you a bit about how these cells work. This is the outer segment, which is the domain of the cell that actually captures light and turns that into electrical signals. That electrical signal now has to propagate through the inner segment, the cell body, and the axon of the cone in order to be fed into visual circuitry. Now, the nature of the signal as it propagates, the way that it is transformed, if at all, when it travels, will dictate the kind of information that's available for giving us our high-resolution view of the world. What is the nature of this propagation? We actually don't know. Okay, what we do know from first principles is that as electrical signals propagate through cables, they tend to get smaller and slower. And if they got too small, that would sacrifice visual sensitivity because you'd get a certain number of photons here and just a tiny electrical response here. And if they're too slow, then they would cause blurring in the image as the world moved and as our eyes move. These slow responses would tend to blur out detail. Okay, so this actually has only been studied once before we got interested. And this was a study that was done in the 90s by Peter Sterling's group. And what they did was they only had the anatomy of the cells. So they had beautiful reconstructions of these cells. So they had the structure of the cable, but they didn't have properties of the cable. So what they did was they borrowed properties from other cells, put them into this structural model that they had, and then asked what happens to signals as they propagate. And what they found, what they estimated, was that signals are attenuated 5-fold as they move from their origin in the outer segment to the terminal for transmission. Now, if you do a few calculations about how big the signal is here, a factor of 5 reduction leaves you with very little here, and it's a mystery how any information makes it to the brain at all. So we became interested in this question of how signals propagate through these cells, it's a crucial first step in how our high acuity vision begins. So the question that I want to pursue with you for the remaining time is, how do signals propagate through foveal cones? And I'd like to point out that this is probably not a problem for peripheral cones. And that's because the peripheral cones are wide, so it's a big highway for a lot of electrical current to flow through. And they're also short, so it's a short road. The current doesn't have long to go from the outer segment where it starts down to the synaptic terminal for transmission. Okay, so this project was initiated by a very talented graduate student in the lab. His name is Greg Bryman, and he was helped by Andy Liu. And we decided that the way to address this question experimentally was to gain access to both ends of the cell and record the electrical activity at the two ends, and then we could deduce what was happening in between, what propagation was like. So we were able to obtain macaque tissue. These animals have a fovea, and we opted to isolate the cells in order to make the two ends accessible. Here's an example of a peripheral cone that's alive, waiting to be examined electrophysiologically. And what you see here is the outer segment where phototransduction takes place, the conversion of light into electrical signals. There's the inner segment cell body, axon, and presynaptic terminal. We can also isolate foveal cones. Here's an example. The outer segment is missing, as it often is, but this should not affect our conclusions, and we can discuss that more if you're interested. You have my email address, and the paper was also published this year. Okay, so the foveal cone lacks the outer segment, but it retains the inner segment, the cell body, this long axon that coils around and then culminates in the synaptic terminal. Now what we do is we put an electrode at either end of the cell. This particular cell has a very long axon that coils up out of the plane of focus and back down again. And by injecting electrical current into one location and then measuring the resulting voltage at both locations, we can get a sense of how signals propagate through these long cells. Okay, there are many technical details to consider. I won't belabor them here. What I will say is that we strive to have our conditions be as natural as possible so that the information that we obtain from these ex vivo experiments, we can relate them back to visual performance, psychophysical and behavioral measures of visual acuity. Now, you can imagine injecting all sorts of electrical stimuli at this end to understand what the cell is doing. And Greg actually injected a whole suite of stimuli. I'll just focus on one. And this is Gaussian white noise. We favor this stimulus because it contains a continuum of temporal frequencies in equal measure. And so we can deliver these temporal frequencies to the cell, and we can ask, what are the frequencies that are preferentially propagated? Why is temporal frequency important? Well, what the cell experiences is some time-varying rain of photons, right? As you look around the scene, the cell will view something bright and something dim and something bright and something dark. And what that results in is a time-varying electrical signal. And so we're injecting a time-varying electrical signal there and then seeing how it's propagated. Okay, this is what the data look like. This is one recording from one cell. There's an electrode at the inner segment that's delivering the stimulus. This is a time-varying electrical current. Scale bar is here. It's scaled to take the voltage of the cell through a physiological range of voltages. Now, this electrode not only injects current, but records it. And so you can see and records the response to that current, which is the membrane voltage. And you can see that down below in this black trace here. Okay, that's the membrane voltage of the cell that results from the stimulus. And then at the same time, we're recording the voltage at the terminal of the cell. And that is what you see in red. Now, the two traces are not identical. There is some change in the signal as it goes through the cell, but this is nowhere near the 5-fold attenuation that was predicted from that computational modeling study that I mentioned before. We can be more quantitative about this by looking at it over we can look at it over temporal frequency rather than over time. And what we're plotting here is the magnitude of the response. And you can see that the inner segment magnitude spectrum, which is shown in black, overlays very well with the terminal magnitude spectrum. they're so close that it's better to look at their ratio instead. So this is the ratio of the response magnitude at the terminal relative to that at the intersegment. And you can see that even for high temporal frequencies, where responses tend to be attenuated the most, we are still above 90%, right? So the fidelity of signal propagation through these foveal cones is excellent. Now, we do the same thing in a peripheral cone for comparison. And as expected from how short and wide this cell is, the terminal and intersegment responses are pretty much indistinguishable, whether you're looking in the time domain here or in the frequency domain there. So let me just show you some group data. So this is our metric for propagation fidelity. You'll see this again. It's the magnitude of the response at the terminal divided by that at the intersegment. And we're doing this calculation for two different frequencies, a low frequency of 1 Hertz and a higher one of 60 Hertz. Okay, so if you look at the peripheral cones in our sample, which are here in brown, and the foveal cones in blue, you can see that the propagation fidelity doesn't fall very far, either at a low frequency or at a high one. And here on the x-axis, this is the length of the cell. There's a systematic variation in cell length with location in the retina. This will become important at the end of the talk. But for now, you can appreciate that even the longest cells in our sample, which would be expected to have the most attenuation, are still propagating signals extremely well. So what is the mechanism of this? How do you get a signal from one place to another without too much of a change? The first thing that came to our minds, and the first thing that most people will think about, is amplification, right? If the signal has a tendency to fall, then you just boost it as it travels, right? This is the basis of the action potential, which carries electrical signals over long distances in our nervous system. So is amplification required? Our amplification comes out of voltage-gated ion channels, so we can block those channels pharmacologically and ask what happens. So we've done a variety of experiments along these lines, and I'll just summarize by saying that, to our surprise, amplification was not required at all. We have no evidence that voltage-gated channel activity does anything for propagation through these cells. So if it's not amplification, if it's not something active that the cell is doing to maintain its signal, then what could it be? It has to be something about the architecture of the cell itself, its behavior as an electrical cable, right? And so what do we have to think about in terms of this electrical architecture? What has three main ingredients? The first one is the specific membrane capacitance. So some of you in the audience might be hearkening back to neurobiology 101 here. The specific membrane capacitance is the amount of electrical charge needed to change the voltage in a unit area of membrane. And the higher that capacitance is, the more charge you need to change the voltage, and the more charge gets soaked up, potentially the less charge is available, the less current is available to change the voltage of the rest of this long cell. Well, what we find is that the specific membrane capacitance has a value of 0.01 picofarads per micron squared, and this is the conventional textbook value. Pretty much any cell that you examine has this value. There are no surprises here. It's a good internal control for our analysis. The next parameter that we have to consider is the internal conductivity. How well current flows down the length of the cell intracellularly through the cytoplasm. And you can imagine that if this value was very high, then current could flow pretty much unimpeded throughout the cell and just change the voltage throughout. That would be excellent propagation. But if the conductivity was low, then current would have a hard time moving around and the cell would not propagate signals well. So what do we see across peripheral and foveal cones? We see that the internal conductivity has a value of 1 microsiemens per micron. This value might be intuitive to some of you. It's certainly not something that I can taste. So let me give you a frame of reference here. So this internal conductivity has only been measured once before for a photoreceptor cell, and that was for a turtle cone. What is that value? It is more than an order of magnitude lower than what we see for primate cones. So the primate cones are doing far better for propagation than this lowly turtle cone, but could they do much better than this? it turns out that we don't think so, right? So we took a beaker of saline that was developed to mimic the intracellular environment of the cell, and we measured the conductivity in this beaker, where there's really no impediment to current flux. And that's the value that we're showing here. So the cones, we know that they contain cytoskeleton and organelles. It's not empty space in there, and yet current can flow through them as freely as in a pool of saline. So we find this to be quite remarkable. Now, the last parameter that we have to consider here is the membrane conductivity, and this is the ability of current to escape the cell into the extracellular environment. And you can imagine that if the membrane conductivity is very high, then current leaks out and there isn't much left to charge up the rest of the cell and change its voltage. So the lower the membrane conductivity, the lower propagation fidelity should be. And this is the first part where we see a divergence between the peripheral cones and the foveal cones of the macaque. Right? We see that the foveal cones actually have a much lower membrane conductivity than the peripheral cones do, which is consistent with what you would want, right? You'd want the foveal cones to retain their current, keep it inside so that it's available to charge up the cell. So what accounts for this difference between the peripheral and the foveal cones? Well, earlier I mentioned that amplification is not required for propagation. That is to say, voltage-gated ion channel activity is not required. And it seems like the foveal cones go one step further than that. They actually downregulate the voltage-gated channel activity in their membranes. And what is a voltage-gated channel? It's a pore. It's a hole in the membrane that lets current go through. And so the foveal cones, by reducing the activity of voltage-gated channels, keeps current inside, right? Excuse me. Now, this is quite the opposite of what we see in most neurons. Most neurons will express voltage-gated ion channels in some abundance in order to generate amplification and push signals along. Foveal cones are doing quite the opposite. They're moving signals over long distances by having a scarceness of voltage-gated channels to keep current inside and allow it to flow throughout the cell. Okay, so the electrical architecture of these cells seems to be quite effective for propagating signals over long distances. But to what extent are they optimal? To what extent are these electrical properties as good as they can be? Okay, so that's the question I want to visit with you next. So how did we ask this question? We did it computationally. We've made all of these measurements, and now we can put them into a model of a foveal cone. We call it the reference cone. And this foveal cone is designed to be among the most slender and the longest in nature. So the most challenging for propagating signals from the inner segment down to the terminal. And now what we do is we systematically vary the parameters of interest. So this is our measure of propagation fidelity, which is the magnitude of the signal at the terminal versus the signal at the inner segment where it begins. And we're plotting that for different temporal frequencies from low to high. And we're plotting that as a function of the internal conductivity. Okay, so here's the natural value that we measure, signified by these blue dots. And then we can make that conductivity lower or higher. What you find is that it's hard to make the performance better by increasing the internal conductivity. The gain here is quite low. And in fact, we can't think of a great way to make it any better because, as I mentioned, it seems to be right up next to just unimpeded ion flow in free solution. So we can't make it much better, but we can easily make it far worse. So here's that turtle cone that I mentioned before, and it's off the scale, right? It's more than an order of magnitude worse in terms of propagating signals. What about the membrane conductivity? Well, we do the same exercise here. And so what you see is membrane conductivity on the x-axis. Fold change relative to the natural value is up on the top, and then the magnitude is shown on the y-axis. Okay, so these blue dots are the the natural values, and you can see that propagation is quite effective. And it's hard to make it much better than it is, right? So we can decrease the membrane conductivity further and we can get some gain, but in fact, it's not practical to go all the way up to this point. In fact, if we block all the voltage gated channels in the cell, all we see is a doubling of the membrane resistance, a halving of the membrane conductance. So we cannot get very far along this curve. We can't do much better, but we could certainly do a lot worse. So if we just put in the values for the peripheral cones, we see that propagation fidelity falls quite steeply. Okay, so it seems like these foveal cones in the primate retina have set up their biophysical mechanisms to be optimal for signal propagation. Now, at this point, we had a question, which is, given that the biophysical properties, the electrical architecture is so finely tuned, would they permit you to have even longer cones? Okay, now this question might strike some of you as frivolous. Why would we want cones to be longer than they already are? But let me explain why we consider this to be an important question. At the very beginning of the talk, I mentioned that the fovea is a tiny portion of the retinal surface area, just 1%. And so what we see is just a tiny fraction of the world in detail. And we have this experience of a seamlessly detailed world because we're constantly scanning our fovea over the scene, and our brain is stitching together those high resolution snapshots. If we could make the fovea bigger, then we would see more of the world in detail at any given instant, and that seems to be advantageous. What do we need to make the fovea bigger? Well, what we need are more cones. So we'd have to stack up more cones in order to expand the fovea. But what you see here is that the inner segments are thinner than the terminals. So that as you stack up the inner segments to make a bigger fovea, that field of terminals is growing at a larger rate. And now what you need are longer axons to connect them. So if you want a larger fovea, you need the axons to be longer. If the axons are longer, what happens to propagation fidelity? What happens to the quality of the signal that reaches the terminal? So we address this by taking our reference foveal cone and making it longer to see what happens to propagation fidelity. And this is a familiar plot now of fidelity at different temporal frequencies. Now this blue bar is the range of lengths of foveal cones that we've seen in our sample, and that represents the range of lengths in nature. And these blue dots represent the foveal cone, the reference foveal cone. Okay, and what you can see is that if we exceed the length found in nature, there's a steep reduction in fidelity. And this is clarified here where we're plotting the first derivative of these curves to show you the rate of change of fidelity. And you can see that the reference foveal cone, the longest foveal cones in nature, are poised right at this point of steep slope. So if we made them even a little longer than they already are, then we would see a drop in propagation fidelity. So we think that this is actually a constraint on how big the fovea can be. You can make it bigger, but then it's less effective because the signals that make it through the cell just are not as good by the time they've reached the terminal. In fact, we think this goes beyond that, and it doesn't just talk about the size of the fovea in the macaque where we made these studies. We think that these biophysical limits may generalize and help to explain this. This is a plot of how big the fovea is across different primate species, from the marmoset, which is about this big, to a human being, which is, as you can see, far larger. And even though the body size is changing dramatically and the retinal surface area is changing by 5-fold, the size of the fovea is relatively constant. So we would argue that one of the constraints on foveal size is the fidelity of response propagation and the limits imposed by biophysics, right? You might think, well, we could tune those biophysical properties to make propagation even better, and that would give us longer cones, but no. The biophysical parameters are already near their optimum. Okay, so let me review what I've told you about in this session. First, I gave you evidence that signals propagate through foveal cones with high fidelity. I suggested that amplification by voltage-gated channels is dispensable. That's unusual. Usually you think about high fidelity propagation as relying on voltage-gated channel activity. I said that propagation in foveal cones is supported instead by high internal conductivity. This also applies to the peripheral cones. What's specific to the foveal cones is a low membrane conductivity. And that seems to rely on a downregulation of voltage-gated channel activity, which is quite the opposite of what you find in most neurons. And finally, I speculated that biophysical mechanisms could constrain the length of foveal cones, and thus also the dimensions of the fovea. So with that, let me thank the people who did the work. All of the experiments that you saw were done by Greg Bryman, this individual here. He was assisted by Andy Liu, who helped us get our primate recordings going in the lab. These are the lab members who gave insight and discussion. And of course, we could not do this tissue. We could not do these experiments without access to very precious primate tissue. And we've received tissue from many generous individuals. One of our principal suppliers is Biomir, and we're very thankful for that. And we're also thankful for our support. The National Eye Institute is our biggest patron. We received invaluable support also from the Bright Focus Foundation and various aspects of Harvard Medical School. So I thank you for your attention, and this concludes the talk.
So Mike, thank you for a wonderful presentation. I think I can speak for myself when I say that I learned a lot and it was really fascinating. So one question that popped into my mind is, it was definitely puzzling that the computational model was practically, 180 degrees different from experimental models. And I was curious if you could shed some light on that, because that's a bit of a puzzling and odd discrepancy. And I was wondering whether the model had accounted for the fact that there was a sparsity of voltage gated channels in the fovea. I mean, are there any reasons you can kind of speculate as to why the computational model was way off base?
Well, Anjali, first of all, thank you for your positive comments on the talk, and I really appreciate the question. That model was wonderfully constructed from the anatomical perspective. So Peter Sterling did beautiful electron microscopy to get accurate measurements of what these cells look like. And this was done in an era where we don't have the high throughput methods that we have now. So it's painstaking artisanal work that he did there. The issue was that they didn't have any of the physiological measurements that we made. And so all they had was the structure of the cell. They just had the shape of the cable. And then they put into that shape the cable properties that had been measured in other cells, whatever was available in the literature. And so it would be like taking a physical cable that you could use to connect two electronic devices together, and not knowing whether the conductive material inside was copper, which is an excellent conductor, or saline or silicon or something that doesn't conduct very well at all. And so in the absence of experiments, they could only infer what the properties of the cable were. So we found that, it's those biophysical details that matter, the actual material. And to go deeper into answering your question would require us to ask, how is it that the internal conductivity is so high in the primate cells? And that requires us to go back to those kinds of anatomical measurements that were made previously and do them with particular attention paid to the organization of structures within the cell, to ask, how does current flow through them, even though they exist? So I think we want to close the loop and go back to Peter's approach now and just refine that with our physiological measurements.
Yeah, that was actually my second question is it because for me, what struck me the most is the fact, as you said, there's a lot of subcellular structures, there's organelles with different densities and different compositions. And it's striking that the current flows right through, like, you know, there's absolutely minimal impedance, which is extremely striking and very different from what you normally see in neurons. I had another, and this was as you were closing up your talk, so it's a little bit more of a philosophical question. Um, so it just struck me how beautifully evolved the phobia is. I mean, this is actually genius if you really think about how nature's evolved it. It struck me, in a very philosophical way. And I was wondering, the, I mean, is there really, any, you'd mentioned about doing, having a bigger phobia to be able to see in much detail. Is that, and it seems like there seems to be some kind of a blocking because, there are limitations as to the terminal. So is that a kind of, in your perspective, an evolutionary checks and balances that's been put in place? I don't know. I was just kind of very curious, philosophically curious about that. Seems like nature set it up perfectly and doesn't want us to fine tune it.
That's a wonderful question, and I have to say that the when you see something like this, first of all, you look at the fovea and it's just overtly beautiful. There's a sort of crystalline nature to its organization that you can just see with a couple of lenses to magnify you can just see it. It's beautiful.
Yeah.
And then when you look down at the biophysical level, where you're thinking about how currents flow through the cells, then you find additional layers that are also beautiful and sensible, and it just makes you wonder, right, and have this sort of philosophical compulsion, desire. What I'll say is that we find specializations at every level. So,
Yeah.
We ask, can you make the cells even longer?
You're right.
Something else that you could ask is, can we make the cells smaller, right? If we made the cones smaller, then they would be smaller pixels and resolve the image at a finer grain. And could we do that? It turns out that that would be counterproductive too. So these cells are remarkable. They actually act as waveguides. So when light falls upon the fovea or any part of the retina, really, the photoreceptors funnel the photons into that outer segment, that region where they can take photons and use that energy to make an electrical signal. And that waveguiding effect only works up to a certain diameter. So if you made the foveal cones thinner, you would start to leak light into neighboring cones, and then you would have a degradation of visual acuity.
Right. You get a blurry, yeah.
That's right. So they're as thin as they can be while retaining this waveguiding feature, and they can't be made much thinner than that. So there are all these trade-offs that are constantly at play, right? There's always a balance. You can have high acuity, but only in a small area. So then you have to move your eyes around the scene. You can make the pixels small, but only so small, right? And so I think it's these sorts of trade-offs that we really want to understand, and
Right.
and they could help us to understand why it is that it's the macula that's vulnerable in macular degeneration. Right? What are the specific trade-offs made there that predispose it to this sort of disease? And then could we leverage that to think about some treatments? And in fact, I have a tendency to give long-winded answers.
No. You're going right in the direction of going of, how this kind of applies to macular degeneration.
Sure. So we don't have, we're looking at things in a very basic mechanistic way now. But if I'm free to speculate, it's actually quite interesting that macular degeneration doesn't attack the fovea first, right? The fovea is a bit protected, and why that is unclear. First of all, I should say that on this topic, everything is controversial, but there's evidence that the fovea itself, the very center of the macula, is slower to go downhill than the rest. And one thing that came to our minds was that if these foveal cones actually have fewer voltage-gated ion channels and less amplification, that actually takes less energy, right? If you're not restoring all of those ionic gradients all the time, then you're less metabolically costly. And so we wonder if that helps to protect the fovea from damage. And if you can downregulate the channel activity out in the more vulnerable areas, I wonder what would happen. So that's just a speculation.
So, I'd love to ask you a dozen more questions, but in the interest of time, let's close this session. And I want to say thank you so much for a fantastic presentation. I think our viewers have learned a lot. And I'd like to tell them that if you'd like to learn more about Mike Doe's research, please visit his lab webpage, sites.google.com/site/Doe Neuroscience. And you can also find his lab webpage through the Harvard Medical School website. So definitely do check out his research and his lab webpage.
Thank you so much, Anjali. I really appreciate the opportunity to talk about my work, and thank you to the